Dr. Hunter is very well respected for his numerous contributions to the study of HIV-1 and other retroviruses. In particular, since the mid-1990s, his group has focused on understanding HIV transmission, in particular in heterosexual couples enrolled in studies in Kigali, Rwanda, as well as Lusaka, Zambia. And this research has greatly enhanced our understanding of HIV transmission from both molecular and genetic perspectives. Dr. Hunter is also highly respected for his dedication to mentorship and capacity building in biomedical research, both here in North America as well as internationally. The title of Dr. Hunter's talk this morning is The Search for an Effective HIV Vaccine, Insights from Studies of Heterosexual Transmission. Dr. Hunter, we'd like to thank you for joining us at CAHR this year in Vancouver, and we look forward to hearing your presentation. Thank you.
Thank you very much. Well, I'd really like to thank the organizers for inviting me to speak today, and also for clearing off the bad weather for at least a couple of hours this morning so I could actually take a run in Stanley Park, which was beautiful, as it always is. It's really nice to be back. So that's my conflict of interest. So basically what I would like to do is talk a little bit about the challenges that virus variation in the cells targeted by HIV pose for vaccine development, very briefly. Then talk about the genetic bottleneck that occurs during transmission, during HIV transmission, and the characteristics of the viruses that initiate infection. Talk about the progress, remarkable progress really, on antibodies that can inhibit HIV-1 acquisition, as well as a possible role for humoral immunity in the natural humoral immunity that develops to HIV in inhibiting reinfection. Then discuss the role of cellular immunity to HIV in containing infection, and finally discuss some recent work that we've both published and some unpublished work of impact of mutations selected by the cellular immune response that they have on replication of HIV and disease progression. Most people at this conference don't need this slide, but I think despite the remarkable progress that's been made, given this rapid rise in the number of cases of HIV across the world, despite the remarkable scale-up of treatment, it's not solving the problem. And as Larry Corey mentioned in his speech yesterday, the likelihood of being able to completely treat our way out of a disease that at least early on is asymptomatic and is sexually transmitted is extremely difficult. And at the current time, even though we've increased treatment to 6 million people, a fifth of those infected in the world, for every 2 people put on antiretroviral drugs, 5 more were infected. So it's a battle that we're losing, and moreover, everyone that's put on drugs has to be on those drugs for life. So a vaccine is really a critical step forward. Our track record, though, has really not been so great. So this is the last 10 years of efficacy trials of vaccines against HIV. In 2003, the VaxGen trial was really based on trying to raise neutralizing antibodies to HIV against the external glycoprotein of the virus, GP120. It failed in protecting against HIV, against acquisition of HIV, and there was no control of HIV. So having demonstrable antibodies to the viral glycoprotein didn't help at all. There was great hope in 2007 for a cell-based vaccine, a cellular immunity-based vaccine. That was the Merck Ad5 vaccine. And unfortunately, not only did it not protect against acquisition of infection, there were more infections in vaccinees than placebo in a certain group of individuals. Not in everybody, but in non-circumcised males, the vaccine seemed to actually increase risk of acquisition. And disappointingly, despite, again, a demonstrable cellular immune response against HIV, there was no control of HIV infection. I think we were all gratified to see the results of the Thai trial, a canary pox and vector-based vaccine with a GP120 protein boost in 2009, that for the first time gave a signal of efficacy, although I think you have to admit that 31% is pretty modest at best. But again, at least it gave us a first positive signal. But unfortunately, again, there was no control of virus replication once infection occurred. I think we're all holding our breath now for this ongoing trial, which is a VRC, Vaccine Research Center for the NIH, which is a DNA adenovirus vaccine that has 3 different envelope components to it. And I think we will hear the results of this efficacy trial now sometime next year. So there are a number of scientific challenges to vaccine development. HIV is a retrovirus. It integrates into the host genome, that it infects, suppresses, and destroys key cells of the immune system, the CD4 T cells. HIV antigens required for protection remain undefined, and specified host responses required for control are really not clearly defined. I think the Thai trial is giving us some hints, but I think we have to go certainly beyond 31% efficacy. And so neutralizing antibodies, cellular immunity, systemic and mucosal immunity are probably all going to be required. The other real issue is that HIV isolates worldwide are hypervariable. And the scale of variation in HIV-1, in a single HIV-1 individual, as you'll see, equals that of annual changes in influenza virus. So the virus is continually a moving target. And there's a short window of opportunity to prevent establishment of persistent HIV infection, because this is a virus that once it gets into the cell and integrates, is going to be there as long as that cell lives. This is an example of the sort of variation that we see in an individual. You'll see a few more what we call phylogenetic trees during my talk. But the basic way to interpret a phylogenetic tree is to go from one tip to the furthest tip, and it's the horizontal distance, and that tells you the amount of variation. So the global influenza epidemic in 1996 had about 8% variation in the hemagglutinin gene. That's about what we see within a single individual after 6 years of infection, somewhere in 6 to 8%. So that's a fairly significant challenge for vaccine designs. But then when we look in Congo in 1997, and this is work from Betty Korber and her group, you see the enormous variation in envelope glycoprotein in each of the different, what we call subtypes of HIV. And so here's subtype A, I've lost subtype B on here. But these are, I guess there weren't any in Congo at the time. But you can see that the enormous variation in the virus. And what we've got to be able to do is to develop a vaccine against all of these if we have a worldwide vaccine. And those subtypes are scattered and differ in different parts of the world. So sub-Saharan Africa is primarily subtype C. South America is primarily subtype B and BF, a recombinant virus. So a number of years ago, when we started this research effort, we started with a hypothesis, and the hypothesis was that an effective prophylactic vaccine against HIV-1 had to protect against the viruses that initiate infection at the mucosal surface, and 95% or 90% of infections in the world are at the mucosa. And the key thing is that we felt that these might be distinct from the bulk of variants that have evolved to survive during their growth in the chronically infected host. And we wanted to ask, what is the nature of these viruses? Because those are really the target of the vaccine. So we really have been aided in this work by a project that my wife started 25 years ago in Kigali, Rwanda. And that was a study of HIV discordant couples. Now, it's not that they can't get on with one another, but they have, one of them has an HIV infection and the other one is uninfected. And what I can tell you is that in Lusaka, Zambia, 20% of couples coming into our testing centers have one partner positive and one partner negative, and it's pretty much evenly split between it being the male partner and the female partner that are infected. After counseling and testing, we see about a 2/3 reduction in transmission. Pretty effective intervention in terms of reducing transmission. And that's because discordant couples, once they learn about the status of their partner, really care about one another and take efforts not to transmit the virus. But for a variety of reasons, a small percentage of individuals do continue to transmit, and we end up with what we call an epidemiologically linked transmission pair. And that represents about 75 to 80% of all new infections in negative partners that we see. That does allow us then to compare the newly infecting virus, the virus that establishes infection in this individual, to the virus in the chronically infected partner, where there's a quasi-species. And one of the first things we found that we reported back now in 2004, time flies when you're having fun, were the following really key aspects of this transmission process. The first was that a single genetic variant from the donor quasi-species initiated infection after transmission. The second was that the envelope glycoprotein, the outer glycoprotein of the virus GP120, had shorter variable loops and less glycosylation, suggesting that there was some selection process occurring that required the virus to change at least it was represented a different virus from the bulk of the population. And those infecting viruses were sensitive to neutralization by donor plasma, or the transmitting partner plasma. So these aren't neutralization escape variants. That was then followed up by a massive study by the CAHR group, headed up, in this case, by George Shaw and lead author Brandon Keel, who looked at about 100 newly infected individuals and using phylogenetic analyses inferred back how many viruses had initiated infection. And in this case, many of these were men having sex with men transmissions. About 80% of individuals that were newly infected, decided had been derived from a single genetic variant, again, entering that individual and establishing infection. We followed up on this with a larger analysis and showed that actually inflammatory genital infections could mitigate this severe genetic bottleneck, and in the context of inflammatory genital infections, larger numbers of variants were transmitted, suggesting that at least part of the bottleneck was at the mucosal surface, or in the mucosa, and similar results were obtained by CAHR. So what do I mean by a single genetic variant? This again is a phylogenetic tree. In green, I'm showing envelope sequences from a chronically infected male partner. This is a male to female transmission pair. And you can see it's very heterogeneous. If we do the horizontal analysis, we start at this virus here, we come back, go to the sort of common node, and then come back out to here. That's about a 6% difference in the sequence of those two envelopes. So there's a very heterogeneous population in this chronically infected partner, but what you can see is very different in the newly infected partner in blue, where these viruses are almost identical, and I can actually tell you that about 65% of these sequences are identical. And we can again infer that a single genetic variant started this because there's a single branch from the donor tree. In instances where we see multiple variants transmitted, we see multiple branches coming off the donor phylogenetic tree. So what's the biological basis of this bottleneck? Could it be that there's limited heterogeneity in the genital fluids of a donor, and that by chance one of these viruses initiates infection, and that by chance in general they have fewer glycosylations? Could it be in the recipient, that selection at the mucosal surface or selection during viral outgrowth? And one could see that if there was a quasi-species of viruses in the genital tract, either male or female, and that a virus had to have a particular trait to be transmitted, that might then give you this clonal outgrowth. On the other hand, if the bulk of the viruses are very similar in the genital tract, the likelihood that one of those would get transmitted in a stochastic fashion would be the case. And so we looked at a total of 8 transmission pairs. I can tell you that it's a lot harder to look at virus in genital tract than it is in blood. This is an example of that, and I realize the people at the back probably can't see this at all. The red dots here are viruses that we've amplified from the genital tract. The blue viruses down the bottom here, the new viruses in the recipient, and this is what we call a highlighter plot, and it compares the consensus of the transmitted viruses, the viruses that initiate infection, to every other virus we look at. And you can see that a majority of these lines have no check marks. Some of them have 1 or 2, and that just shows the homogeneity of the newly infecting virus, and then the heterogeneity of the donor virus. There's a population, the bulk, in fact, of the viruses in the genital tract are, just make sure I stay on time here. The bulk of the viruses in the genital tract are, in fact, clustered in this sort of tree of what we call the genital tract population. But what's striking, and I can show it in a little bit more detail here, is the virus that's most closely related to the viruses that get transmitted is actually genetically unrelated to the dominant genital tract population. And this virus here differs only by 6 nucleotides from the consensus of the virus that gets transmitted. So based on genital fluid sequence analysis from 5 female to male and 3 male to female transmission pairs, we concluded that there is a difference in virus composition in the genital tract and in the blood, that the donor variant that establishes infection is not derived from the predominant genital tract populations, thus limited diversity in the donor genital tract is not the reason for a single genetic variant being transmitted, and these data argue against purely stochastic mechanism for transmission and for selection of a variant with specific traits that favor systemic spread. And Debbie Burris, a postdoc in my lab, published this back in 2011. So this is our sort of current model of what's going on during transmission, that perhaps multiple viruses encounter the mucosal surfaces. Some of them get across and may be defective. Some of them may initiate infection but burn out, or replicate too slowly to get out of the mucosa. And that one virus with particular traits that allow it to establish infection and get out of the mucosa and become systemic is the one that generates the initial clonal breakout in a newly infected individual, and that's in about 80 to 90% of cases. So these viruses do seem to encode more compact envelope glycoproteins with reduced glycosylation. A number of groups have seen that in both subtype C and subtype A, and there's some hint now that it's being seen in subtype B. They require high levels of CD4 and CCR5, the 2 receptors, the primary receptor and co-receptor as they're called for HIV, And that's consistent with these viruses being adapted to rapidly grow in mucosal T cells, which express high levels of both of these co-receptors and are quite distinct from peripheral blood lymphocytes, T cells. They inefficiently infect macrophages, and this was a little bit of a surprise to us when we first discovered this, because I think most people had felt that macrophage infection might be the mechanism to get the virus out of the genital tract into systemic infection. But clearly, these viruses infect macrophages about 100-fold less efficiently than they do CD4 T cells expressing high levels of the two co-receptors. And they may preferentially target alpha-4 beta-7 expressing cells. This is a trafficking marker that targets T cells to the gut lymphoid tissue. And we know that the gut lymphoid tissue is targeted very early in disease, and there's a very rapid depletion of CD4 T cells in the gut within the first 2 weeks of infection. So the genetic bottleneck and systemic spread of a near clonal population provides a narrow window for intervention, plus an opportunity for protective vaccine-induced antibodies to prevent infection prior to a regeneration of diversity. So the neutralizing antibody, developing a neutralizing antibody, a protective neutralizing antibody, is a challenge, however. We know that most licensed vaccines do elicit neutralizing antibodies, and that broadly neutralizing antibodies in humans do exist in chronically infected, about 20% of chronically infected individuals. Moreover, those broadly neutralizing antibodies can passively protect against SHIV challenge. That's a simian version of HIV that has the HIV outer envelope and encodes the HIV outer envelope in non-human primates. And some recent studies that we published this last year really point to development of at least intra-subtype antibody protection over the first year of infection. And when we looked in our Zambia cohort again, the superinfections we observed were all occurring within the first year, and they happened to be occurring in individuals who had very poor neutralizing and binding antibody responses. The problem is that no candidate vaccine in the pipeline currently elicits broadly neutralizing antibodies against HIV. We've seen enormous progress in the area, though, because of the identification of broadly neutralizing, truly broadly neutralizing monoclonal antibodies. And this is work from Dennis Burton's group and from John Mascola's group, as well as others. The VRC identified this VRC01 that neutralized about 90% of viruses, and that was back in 2009. And then the International AIDS Vaccine Initiative group, together with Dennis Burton, identified some novel antibodies that were so-called PG9, PG16, that also neutralized about 80 to 85% of antibodies. This latter group of antibodies actually target the variable loops on the tip of the envelope glycoprotein trimer, and have identified new sites and new targets for antibodies. And then a more recent PGT antibodies actually bind to the V3 loop, which is sort of on the side here. So one of the things that these antibodies do is allow us to think of new ways to target the envelope and new susceptibility points that are shared by many viruses. This just gives an example of how much better VRC01 is than what we considered previously as a broadly neutralizing antibody, B12. The red shows neutralization of strains from a variety of subtypes at less than 1 microgram per mil. You can see that B12 really only neutralized subtype B, whereas VRC01 neutralizes all subtypes and about 90% of them. So broadly neutralizing monoclonal antibodies provide evidence for the existence of shared, highly potent neutralizing epitopes on HIV that could be the target of vaccines. They provide clues for immunogen design. They effectively neutralize transmitted founder viruses that we and others have looked at, and they could provide an interim sort of passive vaccine if we can express them from viral vectors such as adeno-associated virus, and there are some preliminary encouraging results from non-human primate trials in that regard. As Rafi Ahmed, the director of the Emory Vaccine Research Center, constantly reminds me, there's two arms to the immune response. There's the humoral response that produces antibodies. There's the cellular immune response that kills infected cells. And there are two arms for a reason, and that is it's the most effective way to protect. So it's very likely that an effective vaccine will need to also tap into the cellular arm of the cellular immune response. Cytotoxic CD8 T cells recognize and kill virus-infected cells. And we know that HIV-specific CD8 T cell responses have a very early and sustained impact on viral load. They impose the equivalent selective pressure on the virus to highly antiretroviral treatment. A new CTL response, cytotoxic T cell response, can completely replace the viral population with a resistant virus population within about 2 weeks. So that's very similar to what we see with resistance to HAART. And mutation, now it's important though to realize that mutations that allow escape from CTL recognition can relieve this potent suppression of the virus. So just simple mutations will block CTL recognition, but some of those mutations also decrease the replicative capacity of the virus. And just to show you how effective that cellular immune response is, as we see this cellular immune response going up, we see viral load coming down, and the two are clearly linked, because if we remove that CD8 response in a non-human primate model, we just don't see this decrease in viral load. Just for those of you that aren't familiar, the way this cellular immune response works is that viral proteins are digested by the proteasome, and small peptides, about 8 to 11 amino acids long, are transported into the lumen of the rough endoplasmic reticulum, and there they bind the class 1 molecules, the histocompatibility antigens, that can only move out of here once they've bound peptide. They're then expressed on the surface, and it's that complete HLA class 1 with peptide that's recognized by the T cell receptor of a cytotoxic T cell. And of course, the virus can evade this by having, selecting or developing mutations that prevent the proteasome from degrading the protein into peptides, that no longer allow that peptide to fit into the groove of the HLA class 1 molecule, or that alter the interaction with the T cell receptor. So there's ample opportunities for the virus to evade the immune response. And CTL escape mutations can be identified on a population basis by a statistical association of specific amino acid changes with particular HLA alleles, and we call these HLA-linked polymorphisms. We think of them as CTL escape mutations, though. So a cellular immune component of an effective vaccine will provide effective control of virus replication that mimics that of some of our best protective HLA class 1 alleles, such as B27 or B57, that clearly protect an individual and result in much lower viral loads. And they'll target epitopes for which mutational escape results in maximal loss of viral replicative fitness. And we know that, again, B57 is quite effective in this regard. When the virus mutates to escape B57, it generally is much less able to replicate. So one of the questions we asked was, can we identify such epitopes from studies of natural infection? We went back to our discordant couple cohorts, where we know that over time in the chronically infected partner, the immune system selects for variants that are more fit in the context of immune pressure. And then that virus gets transmitted from this, where it's got in this nice, cozy sort of immunologic niche that it's adapted to. It now gets transferred into a new individual that in most cases is quite different immunogenetically, and the virus now has to cope with that. So we have asked how frequently do these mutations get transmitted, and I won't talk about that today. We asked what impact might they have on early set point viral load in the newly infected partner. Can we correlate CTL escape mutations with viral load in this individual? How do they affect the replicative capacity of the virus? And how might they also affect long-term pathogenesis in this newly infected individual? So in other words, does the immune response in this individual imprint the virus in a way that modulates how that virus is going to initiate infection and induce pathogenesis in the newly infected individual? So the first hint of this really came from some work that we did with Paul Getford back in 2008 in this JXMed paper, in which we showed that increasing numbers of CTL-linked, HLA-linked polymorphisms in the GAG gene, and that's in the GAG protein, that's the internal structural protein of the virus. It has to undergo multiple interactions with other GAG proteins, and so has a very constrained structure. That increasing numbers of mutations resulted in a lower viral load, and that was almost a log when we compared transmission of viruses with more than 9 CTL mutations versus those that had less than 3. And so then we asked, well, we would expect that what this is doing is interfering with the replicative capacity, the ability of the virus to reproduce when it gets into a newly infected individual. And so we've asked, to what degree does the viral replicative capacity defined by the GAG gene of the transmitted virus contribute to the set point viral load and early pathogenesis in this newly infected individual? And to do this, we actually tapped into a methodology, a sort of modified methodology that Mark Brockman and Sabrina Broom here at Simon Fraser University had developed. And we went to 149 transmission pairs. I should say we, it's the classic PI's we. My students, Dan Claiborne and Jessica Prince, cloned 149 GAG genes into this MJ4 infectious provirus. It's a subtype C provirus, and these are all subtype C viruses. We can then transfect that provirus into 293 T cells, a very easily transfectable cell, harvest virus from those cells, titer the virus, and then put it onto T cells that Mark developed, and monitor the spread of the virus through the culture. And we see dramatic effects on the replicative capacity of the virus based on just introducing different GAG genes into that virus. And we published this recently in PLOS Pathogens. I won't go over all of that data, but the two things I think that we really came away with was that the viral replicative capacity that's conferred by the transmitted GAG is positively correlated to the early set point viral load in newly infected recipients. So the better the virus replicates in this assay, the higher the viral load that we see in the individuals. Although that is clearly modulated by a number of host cell factors. And again, last year we published on, the Zambian cohort, where we showed that gender, early protective HLA alleles, as well as HLA sharing between couples, have dramatic effects on viral load. So if you get a virus from your partner and you share a single HLA-B allele with that partner, your viral load on average will be almost half a log higher. What was a real surprise, though, was that transmission of low viral replicative capacity viruses was associated with a slower decline in CD4 counts compared to transmission of viruses with high viral replicative capacity. And that indicated accelerated disease progression in individuals infected with high VRC, or viral replicative capacity viruses. The problem was these initial published studies were based on only 63. I mean, it's remarkable we got the p-values we did with just 63 epidemiologically linked recipients for which longitudinal CD4 counts were available. And Dan and Jessica went back and identified another 64 acutely infected individuals for which we had longitudinal viral load, and subsequently determined the viral replicative capacity for all 127 and reanalyzed this relationship to early pathogenesis. And I think you can see this in a Kaplan-Meier curve here. This is viruses with the first tercile of the poorest replicating viruses, the third of all of these viruses, around 40 viruses, 42, that have the slowest replication in culture. And then these are the two other middle and upper terciles that replicate much better in culture. And now we're able to, because of an expanded data set, go out to almost 6 years of study. And you can see all the way out to 6 years, there's quite a dramatic difference in the decline in CD4 counts, just based on in vitro replicative capacity. Whoops, sorry. At less than, that's going down to a CD4 count of less than 350, which is the UNAIDS recommended treatment level. If we look below that at less than 300, it's even more dramatic. And because the upper two tertiles pretty much track together, we basically then combined those for studies of less than 200, and you can see that the lowest replicating viruses and the higher replicating viruses, again, differ significantly in their progression to what we would classically call AIDS. I'd already mentioned that replicative capacity correlates with viral load. Is this just simply a reflection of viral load? And I think I'll have to move quickly, but the answer is no. We see very significant, this is looking at down to less than 300. We see a very significant effect of replicative capacity alone, of viral load alone, but when we do a Cox proportional hazard model, taking into account either viral load or replicative capacity for log viral load, both remain highly significant. Whoops, sorry, I keep hitting the wrong button here. Both remain highly significant with quite significant hazard ratios when we take each of those into account. Sorry, that is 350. At 300, it becomes even more significant, and again, both values stay independent of one another. So what it says is that there's two different effects, two different components are affecting CD4 decline. There's something linked to virus replicative capacity, and then there's the ability of the immune system to ultimately control viral load and hold that virus in check. But both of those are doing something different, and both impact CD4. And in fact, you can see that very nicely here, where again, we see an independent but additive effect of replicative capacity with viral load. And this is, the green is low replicative capacity, low viral load. Low viral load, high replicative capacity, and look at the difference. And then the red is showing high replicative capacity, high viral load. So it clearly stratifies those with intermediate viral loads, dramatically altering disease progression, and individuals with high viral load demonstrate a loss of control and are associated with a rapid loss of CD4 cells. So, in conclusion, the replicative capacity of a transmitted virus, as defined by a GAG sequence, dramatically impacts the course of HIV pathogenesis in that individuals exhibiting viral variants with low viral replicative capacity exhibit a delayed CD4 T cell decline all the way out to 6 years. This observed effect appears to be independent of both the influence of set point viral load, which is known to correlate with CD4 decline, and we've also looked, given the larger numbers now, at other host factors such as protective HLA alleles, which are going to be able to allow control of viral load and that are known to modulate viral control. And in fact, we see complete independence of the protective effect of B57, B5801, and viral replicative capacity. So transmitted viral characteristics as well as host immunogenetic factors clearly play significant and independent roles in the trajectory of HIV pathogenesis. And what we think is going on is that there's something during the first 2 weeks of infection prior to the appearance of the adaptive cellular immune response that allows control of viral load that is setting the trajectory of HIV disease. One possibility is that in viruses that replicate rapidly is that there's a more complete depletion of the central memory pool of gut-associated CD4 T cells prior to immune control, or that there's increased early immune activation with an exacerbated inflammatory response and bacterial translocation. Bacterial translocation is known to occur because of HIV infection, and that sets up a continuous inflammatory immune activation response, and that's known to be involved in CD4 decline. Or a combination of these two things may very well be occurring in viruses that replicate very well and is occurring to a much lower extent in individuals infected with viruses that replicate poorly. I think the hope is that if we can develop a really strong CTL response in vaccines, that even if acquisition fails, even though if we fail to prevent acquisition, a vaccine that can reduce virus replication during this early stage of HIV-1 infection could clearly have long-term benefits in terms of disease progression. And I think in some of the monkey models where very strong CTL responses have been generated, but acquisition has still occurred, we've seen much longer and infections and much less progression in those individual monkeys. So I think this is a real thought for the future. So in conclusion, sort of future directions, recent progress in the field has created a renewed sense of optimism for AIDS vaccine development. The focus is on development of a safe, effective vaccine that prevents and controls HIV infection, and the hypothesis that this will require induction of both broadly neutralizing antibodies and cell-mediated immune response, and maybe also binding antibodies, as has appeared to come out of the TIE trial. The major immunogen design challenges remain, however. Broadly neutralizing antibodies for prevention of HIV acquisition are still an elusive target for immunogen design, and we still need to understand what is truly an effective cellular immune response for control of HIV that can be elicited by a vaccine, because the Merck trial obviously did not control. However, I think our recent studies show that a vaccine that generates an effective cellular immune response could reduce the replicative capacity of subsequent transmitted viruses and impact disease progression in acutely infected individuals. Projects like this require multiple collaborations. I have to really say that the EVC has been an amazing place to work. Dan Claiborne, Jessica Prinz have done the bulk of the work I talked about just recently. Melinda Schaefer and Ling Yu have also been involved. Cynthia Durdeen really started a lot of this work, is now a colleague of mine at Emory, and her group and mine continue to collaborate very closely. We've got a great biostatistician. We couldn't do any of it without the Zambia Emory HIV research project headed up by my wife, Susan Allen. We've got some wonderful collaborators there that have headed up the IAVI Protocol C program that allowed us to do the longitudinal CD4 follow-up. And Jill Gilmore, Manuel Cormier, and Matt Price at IAVI have been wonderful collaborators. Mark and Sabrina, we've been really working closely with Jonathan Carlson and David Heckerman at Microsoft Research in analysis of our sequences, and I continue to collaborate with Paul Gepford on immunology and Dick Caslow and James Tang on immunogenetics at UAB. Have to thank funding, and I think if you'll notice here that the Canadian government has been very generous in helping my wife maintain some of our studies in Zambia, and we're very appreciative of that. Thank you. I'll stop there.
Thanks, Eric.
Okay, thank you.
Just hold on a second. Well, thanks very much, Eric, for that wonderful talk. I think it's really important that highlighted the importance of the discordant couple, especially a discordant couple in Africa. I think that's a very powerful model. And not only, we brought you here for 3 reasons. One is for the quality of your science, which we just saw today, and I think that speaks for itself. The second was the clarity of your ability to communicate, and in a forum like this, that was really important. And then the third reason is because you're a very approachable person. So I'd encourage everyone at the gala tonight to approach Eric and tell him all about your work and ask him all the different questions that you may have. So on behalf of everyone in CAHR, I want to thank you very much for doing this for us, Eric. Thank you.
Thank you.